This is a continuation of my discussion about nucleic acid structure. And in the previous recording, I have shown to you that the way that we assemble the nucleotides makes it so that we have a sugar phosphate backbone right here, and the bases protrude. And that if ever we have two strands, okay, they form a ladder shape, or in most cases, they form a helical or a something like a spiral staircase shape, which I will try to elaborate further here. So, before we do that, we have to recognize that there are intrinsic differences between RNA and DNA, of course, as it should, because other than the first letter, R and D, they have a lot of things that are very you could contrast with each other. And that would be more obvious when we discuss the steps in the central dogma. But for now, as far as the structure and the composition are concerned, we could first, one, compare their sugars. And this is the easiest because that's what makes their first letter different. The R in RNA stands for ribose. That's why we call this ribonucleic acid. And then for DNA, of course, D here stands for deoxyribose. So DNA stands for deoxyribonucleic acid. Okay. And I believe I have already discussed the difference of these two sugars in the first recording I had on nucleotides. Now, normally RNA is single-stranded, so and when I use the word here, usually that is, of course, with due respect to some RNA strands that are double-stranded. But as far as the central dogma is concerned, the steps that we will be discussing about how RNA and proteins are synthesized, we usually see RNA existing as a single strand. And for DNA, although of course there is a possibility of finding single-stranded versions of DNA, for most part, when we talk about DNA and the central dogma, we imagine it as a double-stranded molecule. Okay, and as far as the bases are concerned, RNA uses the bases adenine, guanine, cytosine, and uracil. For DNA, we have actually almost the same, except that RNA's uracil has been replaced with thymine. Okay, and we could kind of say that uracil and thymine are mutually exclusive. The fact that the moment you see uracil or letter U in any sequence of letters, that will tell you that we have an RNA strand. But if you see T in a sample, if it's a undegraded and unmutated sample, it's that of DNA. Now, it may be possible to see a strand wherein there's a mixture of uracil and thymine, but that is odd. Usually, that kind of suggests to you that there's some kind of mutation or something unexpected that happened because you're not supposed to see them at the same time. Okay, in uh, in normal genes or nucleic acids. But we will talk about mutation in another discussion. But for now, other than knowing the differences of the components of RNA and DNA, we also have to recognize that just like proteins, nucleic acids have their own levels of organization. Now, if you are still, by the time you're watching this, familiar with the levels of organization for proteins, it's quite convenient because we have some similarities in the levels of organization for nucleic acids. For example, when we were discussing proteins before, the primary level is about the sequence of amino acids. And here we use the same word. For nucleic acids, their primary level also involves the sequence. Of course, in this case, we're not anymore talking about the sequence of amino acids, but the sequence of the nucleotides, or at least the nucleotide bases. And of course, just like what I discussed in the previous recording, the nucleotides are linked together by what we call as the phosphodiester bond. So the only bond that matters in the primary level of organization for nucleic acids are the phosphodiester bonds. When we reach the secondary level of organization, there now starts to become interactions between bases. Okay, so for example, adenine could interact with thymine, cytosine could interact with guanine, and the interaction that they make is primarily hydrogen bonding. Okay, and it would take me time to draw, like, justify that it's possible to form hydrogen bonds, but well, maybe I could try to draw one. Think about it. Maybe what my let me, I think, draw cytosine. Yeah. So here I'm drawing cytosine. And if you think about this, remember, cytosine has quite a lot of NH groups, right? Wherein we know that in an NH group, we have the hydrogens having partially positive charges. And then I have here a carbonyl group, which actually is present in all our pyrimidine bases, okay, and almost all of the bases, which can have a partial negative charge. And remember, these things are the requirements for hydrogen bonding. So if you could just imagine cytosine and probably another base in front of it interacting via these partial charges, that justifies that it's really possible to find H bonds. Remember, all other bases have, at least one or two of these NH groups. Okay. Now, in the secondary level, of course, it will differ depending on whether it's RNA or DNA. If it's an RNA molecule, since most RNA strands are single-stranded, that means that it will have H bonds intramolecularly. So, like, it forms hydrogen bonds within itself because it's only one strand. Okay. And for DNA, since usually DNA involves two strands, then you could just imagine those two strands um, forming hydrogens between each other. So you could say that the bonding is intermolecular from one strand interacting with another strand. Pretty much what I showed here. So if this is like strand number one, and this is base from strand number one, and this is a base from strand number two, and it's possible for these two bases to form a hydrogen bond, then of course we say that this hydrogen bond is intermolecular. So that is something that you would see in the secondary level for usually for DNA. Okay, since again it's the DNA which is usually double stranded. Now, do note that if I have the secondary level for DNA strands, and I did mention we have here the formation of ladder-like figures, other intermolecular forces, primarily hydrophobic forces, or like London dispersion forces, and etc., could actually allow the conversion of the structure to something twisted, or even we could say helical. So it's actually wrong to think that by default, our strands are like flat ladders that you usually use to, uh, to climb up stuff, right? No, it's not a straight ladder. It's, well, if you want to stick with the analogy of a ladder or stairs, it's more of like a spiral staircase instead of a straight, you know, set of footsteps. Okay, or sorry, wait, stair steps. I mean, and therefore, we recognize that every DNA molecule has a certain degree of coiling. Okay, so not too loose, not too tight. However, if I have the tertiary level, we could imagine that normal coil encountering further interactions. For example, it is possible, of course, depending on many factors, for this strand to supercoil. This is usually in the presence of, or in the case of prokaryotes, wherein, if this is coiled like this, supercoiling means that it becomes even more coiled. And of course, this isn't like the most accurate depiction of what's going on in real life, but you could just imagine that this is more tightly coiled compared to this one. So that's one way you could imagine supercoiling. And then plasmids, which are circular pieces of DNA, could also be present, and that's for prokaryotes also. So instead of thinking of a helix as just like going from top to bottom like that, you could just imagine that, you know normally we go bottom, but then it goes full circle back to the starting point. So from something going in a linear fashion, it could now become circular. So that's usually what we call as classmates. So these are, quote unquote, further interactions, which apparently is more commonly found in prokaryotes or bacteria. But for the quaternary level, it involves binding to other molecules, and usually we're talking about proteins here. For example, histones. I almost stopped the slides. Anyway, for example, histones are proteins which normally form complexes with DNA. So it's as if histones are like globular proteins. You could just imagine our DNA molecules, all of those millions of nucleotides wrapping themselves around a histone. And that actually allows our DNA and our genes to be compressed a lot, allowing them to be very, 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 very compact. And that should make sense because for us eukaryotes, remember that our cells have nuclei. And since our DNA are supposed to be found inside our nuclei, uh, they should fit. Remember, a nucleus is not that big compared to the entire cell. So if the DNA is to fit inside the nucleus, then it should be in the compact state. Therefore, that's why we could find our DNA wrapping themselves around histones. And probably they're just out there waiting to be, you know, to be removed from that coil or from this wrap around, uh, like waiting to be released in a way. But of course, that is another complex thing that we cannot discuss in detail here. But all you need to know is that quaternary level involves additional molecules like histone proteins. Now, it's not just enough for us to know about the four levels of organization, because if we are to study the central dogma or the processes that are essential for the expression of genetic information, we must further dive into the details of what we call the DNA double helix. Okay, which again normally is under the secondary level of organization, but that is the functional form, the way that we see DNA being used to make RNA, which later on is used to make proteins. We start with a double helix and we should understand its properties. Of course, it wouldn't. I think it would be weird if I do not mention the names of significant people related to the discovery of the double helix, as important as this discovery is. So, of course, we attribute it to Watson, Crick, Franklin, and Maurice Wilkins. And I'll just write their names like all of them are equal because there's a lot of history with these people. Okay, but these are four people that you have to know, or at least remember when you think of the double helix. So, by the contribution of these people and more, what do we now know about the double helix? One, since we have in the double helix two strands, it would be helpful for us to first recognize or realize that those two strands are identical, meaning the sequence or the arrangement of the bases from 5' to 3' end of one strand is exactly the same as that of the other. Okay. Second, there should be what we call base pairing. Okay. And there are two popular base pairs. Sometimes we call them the Watson-Crick base pairs, and those are AT and CG. That is, adenine only base pairs itself with thymine, and thymine only pairs with adenine, nothing more. Adenine cannot, base pair with C or G. It cannot even base pair to itself. The only one that we usually find in DNA paired to A is T. And the same goes with C to G or G to C. Now, if we are to establish this rule, then, or this admission that this is the only pair for A and T, and this is the only pair for C and G, we could understand the Shergaff's rule, which states that the percent of A is equal to the percent of T, because again, if they are to pair with each other, then how much A should be? It should be equal to how much of T is there, right? And then the same goes with C and G. They should be of equal percentage. Now, of course, since these are the four bases that we find in DNA, only the four of them, it should be expected that in a normal, undegraded, okay, unmutated DNA sample, so if it hasn't been like destroyed or whatever, all of these should equate to, or all of these should add to 100%, right? They are all of the bases present. Now, if we confirm that all of them are equal to 100%, we could try to rearrange these things and come up with the equation that the percentage of the sum of A and G is equal to the percentage of the sum of C and T. Now, remember that adenine and guanine are the purines here. Cytosine and thymine are the pyrimidines. So it's like saying that according to the Chargaff's rule, or sometimes we call it the Chargaff's first rule, the percentage of purines is always equivalent to the percentage of pyrimidines in a normal unmutated DNA sample. So, for example, how can that help us? Well, of course, maybe it would make sense if I mention um, a test question. So maybe if you are asked, let's let's say that hypothetical DNA sample has been discovered to have 12% adenine. How much percent of um, cytosine is there? So how do we solve something like this? Based on the Chargaff's rule, we could start with plotting 12% on adenine. So if the adenine percent is 12, what does that mean? Since A is equal to T, then if A is 12%, then T should be also 12%. Now, what does that give us? Remember, if I add A plus T plus C plus G, that should be 100, right? And since, A and T are both 12, so that's like, this is 12 plus 12. So this is 24, at least in my problem here. Then plus C plus G equals 100. So that means that if I transpose 24 to the other side, C plus G is equal to 100 minus 24. So 100 minus 24 is 76. Now, remember that since C is equal to G, we could imagine as if, if they are equal, then this is like, we could think of them as like x plus x, right? Because they're the same. So x plus x is 2x. So basically, to solve this, you have to divide both sides by 2. X is equal to 76 divided by 2, or 38. That means that G is 38%. That's one of the two x's, right? And then the other one, of course, is C, which is 38%. And therefore, the question mark percentage C is 38%. So of course, this is again assuming that the DNA sample is perfect. It has no mutations or errors. So um, the Chargaff's rule allows us to discover the percentage of all bases just by finding out the percentage of one of them. Okay. Now, another property of our DNA double helix is that the strands should be antiparallel. If you remember, in the previous recording, I already mentioned that word. That when we say antiparallel, two lines go in opposite directions, like so. Since here I did mention in the previous recording that 5 prime to 3 prime is like this arrow, and then here 5 prime to 3 prime, notice that it's as if I always start at the 5 prime, get used to that, goes in this arrow. So anti-parallel in that sense, right? But of course, the question is, why? I mean, why don't we do parallel? Is it unacceptable? Let's see. If you do parallel, what would happen? Remember, your two strands are identical. So if I have a very short strand, 5 prime, AGCT 3 prime, just four bases. And then parallel means they both go in the same direction. So all I have to do is to copy paste this strand, and then I have this. If I have a parallel double helix, look at the bases that would be facing each other. A will face A, G will face G, C will face C, and so on. And remember, I said a while ago that base pairing never involves pairing of A to itself or any other base to their own self. So this is unacceptable because these base pairs don't make sense. We don't have T T or C C base pairs. There's nothing like that. So we violate the base pairing rule if we follow the parallel arrangement. So by anti-parallel, it means that if this is the first strand, I have to start the second strand at the opposite part. So I'm gonna write this first, 3 prime, and then this first, and then followed by this, this, and then 5 prime. And now notice by doing that, I now get, like, look at this, 80. That's a base pair. GC or CG. That's the base pair we're looking for. And then finally, another round of 80. So it actually makes sense that the reason why I mentioned in the previous recording, we have to follow the anti-parallel arrangement is basically for us to follow the normal Watson-Crick base pairs. Okay? Otherwise, they won't be able to complement each other very well. Okay? And sometimes, if you try to combine these two ideas, that it should be anti-parallel, so that it could base pair properly, or in order to base pair, it should be anti-parallel. Sometimes we use the word complementarity. That in order for the two strands to be complementary to one another, they should have this set of pairs, and they should have the anti-parallel arrangement with respect to one another. But another property that's essential for the double helix is that we recognize its handedness. Remember, I did mention that instead of thinking of DNA as like a ladder, we should imagine it as a spiral staircase. But now that leads us to another question. Isn't that when you spiral, you could spiral either clockwise or counterclockwise? And apparently, that actually matters because if you spiral in the right-handed fashion, that means you spin clockwise from 5 prime to 3 prime. So clockwise is like this, right? Um, you will have two major types of DNA, which apparently are the active forms of DNA. By active, they are the ones that participate in the central dogma. Those are the aDNA and the bDNA. Now, the details about these two are quite complex because they have slight differences in as far as their height is concerned, and probably their thickness. It's kind of advanced, more advanced than I would like to discuss this recording. So maybe just to give something significant, maybe it would make sense, or it would be significant for me to mention that the bDNA is the most common form of DNA. That most of the time, the DNA that we study in the laboratory or observe during research is the B form. The aDNA is less encountered, is more of a special case. And in fact, it's the bDNA that allowed the discovery of the actual double helix by these people up here. Okay. Although, of course, these people also found this, but this wasn't the critical form of DNA that led to the discovery. Now, in contrast to the right-handed DNA, the left-handed, which means that our helix goes in the, sorry, that's clockwise, in the counterclockwise fashion, is the inactive state. And in fact, we have only one example of that. It's the zDNA. Z here actually stands for zigzag. So unlike A and B DNA, the Z here actually has a meaning because, of course, it's not a perfect zigzag, but the way that zDNA looks is rougher on the edges. So something like this instead of something smooth like this one. And just like the aDNA, the zDNA is normally uh, rare to find. Okay, and uh, going deeper, zDNA actually has some purpose. However, it's not just like aDNA as common as the bDNA. So these are the properties of the double helix that we need to know as we try to understand more about nucleic acids in the central dogma.